Let's move on to the next question. Question number 54. Structures which constitute the triad in skeletal muscle contraction are. So, let us learn about skeletal muscle contraction now. So, let us deal with the neuromuscular junction now. What is the neuromuscular junction? The junction between a motor neuron and the muscle fiber is called as neuromuscular junction. And let us learn about the events that take place in the neuromuscular junction. So, what are the events that take place in the neuromuscular junction? So, what is the first thing that happens? Arrival of an action potential at the presynaptic neuron. So, for this to occur you need voltage gated sodium channels. So, first step is arrival of action potential at the presynaptic neuron. And what does this action potential do? It opens voltage gated calcium channels at the synaptic knob or synaptic terminal, and calcium enters inside. What does the entered calcium do? Whenever calcium is released, it should cause a movement. So, what is the movement here? It moves the vesicles towards the axon terminal or the synaptic membrane. So, number 1 action potential, number 2 calcium. Whenever you have calcium you should have a movement. What is the movement here? Movement of acetylcholine containing vesicles towards the membrane, and acetylcholine is released. What does this acetylcholine released do? It attaches to the nicotinic receptors present on the postsynaptic membrane. Nicotinic receptors. And what does this do? It increases the sodium influx by opening voltage gated sodium channels and thereby development of an action potential again. So, this action potential travels along the sarcolemma like this and moves along the T tubule. So, these are the T tubules here, and the action potential moves along the T tubule like that and it travels into the muscle fiber. And what is this portion? See, this is the structure of a myofibril. Okay, this is a myofibril which is encircled by sarcoplasmic reticulum. So, the T tubule along with the adjacent to sarcoplasmic reticulum is called as a triad. So, what is a triad? The T tubule along with the two adjacent sarcoplasmic reticulum is called as the triad. So, what happens in the triad when action potential travels down the T tubule and reaches there? Let me enlarge this portion. So, there are two types of receptors called dihydropyridine receptors and ryanodine receptors. What are these receptors? See, when action potential travels along the T tubule, there is a conformational change in the dihydropyridine receptor and it attaches to the ryanodine receptor. There is a physical contact between the dihydropyridine receptor and ryanodine receptor, and there is release of calcium from SR. Calcium is released from sarcoplasmic reticulum. This released calcium in turn acts on the myofibril and causes myosin to slide over actin, causing power stroke and thereby muscle contraction. See, if you see in the presynaptic neuron, first step was action potential, second step was calcium, and third step was a movement. What was the movement? Movement of vesicle towards the membrane. Similarly, in the postsynaptic muscle also, first step is action potential, second step is release of calcium, and this calcium here also it caused a movement. What is the movement? Movement of myosin over actin. So, action potential, calcium, a movement. So, this is a sequence here. So, structures which constitute the triad in skeletal muscle, sarcoplasmic reticulum and T tubule. So, both. Lambert-Eaton syndrome is. So, let us learn about the disorders of neuromuscular junction. Okay. So, let us start with the voltage gated sodium channel. Blockers of this sodium voltage gated sodium channel are tetrodotoxin, which is present in puffer fish. It is a voltage gated sodium channel blocker. Autoantibodies against voltage gated calcium channel is called Lambert Lambert-Eaton syndrome. And for the movement of vesicles to the membrane, you need certain proteins called as snare proteins. What are snare proteins? There are three types of snare proteins: synaptobrevin, syntaxin, and SNAP25. These are important snare proteins, and these proteins are important for the movement of acetylcholine vesicles towards the membrane. And what is the toxin which causes damage to these proteins? Clostridium botulinum. There are different types of Clostridium botulinum. Clostridium botulinum B blocks brevin, see B for B, and Clostridium botulinum C blocks syntaxin, syntaxin for C, and A and E blocks SNAP25. So, snare proteins are damaged by Clostridium botulinum. See, when snare proteins are damaged, acetylcholine cannot be, the vesicle cannot move towards the membrane, and acetylcholine cannot be released. When acetylcholine is not released, what kind of paralysis it causes? Yes, it causes flaccid paralysis. It causes flaccid paralysis. Clostridium botulinum causes flaccid paralysis. What about Clostridium tetani? Clostridium tetani blocks GABA and glycine, which are inhibitory neurotransmitters. When inhibitory neurotransmitters are blocked, what happens? There is excess acetylcholine, and this causes excess contraction, especially masseter is affected. This is called lockjaw. So, Clostridium botulinum causes flaccid paralysis, whereas Clostridium tetani causes spastic paralysis. Mutation of ryanodine receptor causes malignant hyperthermia. See, what happens when there is a mutation of ryanodine receptor? It becomes hyperactive and it releases more and more calcium. So, there is more muscle contraction and more heat production, which causes malignant hyperthermia. So, these are all the disorders of neuromuscular junction. So, the 55th question, Lambert-Eaton syndrome is autoantibodies against calcium channels. 56th question, malignant hyperthermia is due to mutation of ryanodine receptors. 57th question, Clostridium botulinum causes flaccid paralysis by blocking snare proteins, especially Clostridium botulinum B blocks synaptobrevin. What is the mechanism of Clostridium tetani? It blocks GABA and glycine. Tension developed by passive stretch of muscle is called. See, let us learn about the different types of tension produced in the muscle. How can you increase the tension in the muscle? One is by myosin moving over actin and causing contraction of the muscle, which increases the tension in the muscle. So, this is called active tension. So, the red one is called active tension. And the other way by which you can increase the tension in the muscle is by stretching the muscle. Imagine you stretch a rubber band, the tension increases. So, this is called passive tension. The blue one is called passive tension, and the green one is the sum of active and passive, which is called as total tension. This particular length at which active tension is maximum is called as the resting length or optimal length. Optimal or resting length. See, if you see the graph, at lengths lesser than the optimal length, the contraction decreases. At lengths more than the optimal length also, contraction decreases. Why? See, if suppose this is the optimal length, so this is the length at which the myosin filament slides over the actin like this, and there is a power stroke and it causes contraction. At lengths lesser than this, myosin can touch actin, but there is no space for actin to move, so there is no contraction. At lengths more than this, what happens? Myosin cannot touch the actin at all, so there is no contraction again. So only at this optimal length there can be a sliding filament theory and power stroke, which will cause a contraction and increase in tension. So the tension developed by passive stretch of muscle is called, so the curve C gives the passive tension curve.